Hello everyone, my name is Wend Yam Davy Ouédraogo and I'm a PhD student at the Laboratory of Computational Biology in the Department of Computer Science of University of Sherbrooke. I'm working on comparative genomics and phylogeny. I would like to present you my recent poster for TranscriptDB. TranscriptDB is a transcript-centric database developed in my laboratory to study transcript conservation and evolution within gene trees. So, the interest of ontology between genes has led to develop several genomic databases. They provide data and methods on gene evolution. We can retrieve the information about the different types of relationships between genes like ontology and paralogy. Otherwise, they do not provide information on transcript evolution like information about splicing ontology and transcript tree. Eukaryotic genes can produce several distinct alternative transcripts with different functions due to alternative splicing. Alternative splicing is a mechanism that makes the transcript a refined level of functional homology relationships. So, why do we need a transcript-centric database? More and more data are available in open source databases, and the information on their evolution can be useful to reconstruct accurate gene trees. More knowledge about transcripts can benefit to assess the evolution of genes. And gene analysis at the level of transcripts allows to account for all products of a gene. What is the transcript database content? TranscriptDB provides types of homology relations between transcripts, also transcript trees and gene trees. It also provides comparative genomics data display, tools for data analysis and processing, and finally data export and a programmatic access API. TranscriptDB allows you to download data in FASTA format or CSV format. The collected and computed data are loaded into a custom PostgreSQL relational database. The database provides relevant clusters of conserved transcripts, and transcript phylogeny is computed, thanks to a new transcript similarity measure. We developed two algorithms, respectively a clustering algorithm to get conserved transcripts and transcript phylogeny algorithm to reconstruct transcript tree. In this purpose, we developed the transcript similarity measure that is the half of sum of two scores called respectively deg-om and exon-om. The deg-om accounts for the quantity of nucleotide shared, while the exon-om accounts for the quantity of exons blocks shared. The transcript phylogeny algorithm takes at the input the cluster of conserved transcripts here from TranscriptDB clustering, and the corresponding gene tree give at the output a transcript tree. The algorithm has two steps. The first step is the transcript subtree construction, and the second step is the transcript supertree construction. So the first step construct the transcript subtrees in congruence with gene trees for each transcript cluster resulting from the TranscriptDB clustering. Then the second step is to construct a transcript supertree by combining all subtrees based on the transcript similarity measure and reconciliation with gene tree. The resulting supertree is used to infer homology relationships between transcripts. What type of data can you get in TranscriptDB? We can get transcript level resources like transcript clusters, transcript trees, and stable ID mapping for transcript trees, and the decomposition of exons blocks and the conservation scores. Also, we can get gene level resources like gene splicing structure and the set of exons. We can get also gene trees and the types of homology relationships between genes and the alignment of all homologous genes. To conclude, we presented a transcript-centric database and a new method based on splicing structure to compute clusters of conserved transcripts to the reconstruction of transcript and gene phylogenes. The database provides tools for analysis and visualization of genomic data. A future work will be to evaluate all conserved transcripts that are present in TranscriptDB. The database content is accessible through a REST API, and the tools is freely available through its web interface at www.transcriptdb.copious.usherbrook.ca. I would like to thank the Canada researcher, the University of Sherbrooke, and my supervisor and all the members of Copious Laboratory. It was a pleasure to present our research to ISMB 2022. Thank you.